Hello and welcome to our poster. Thank you for stopping by. I'm Manuela. I am a bioinformatics data scientist at Benevolent AI, and I work in the precision medicine team on transcriptomic data. Our goal is to uncover the mechanistic driver of disease to develop the right medicine for the right patient. I will present recent work our team has conducted aimed at benchmarking algorithms to identify clinically relevant subpopulations of patients. Our work stems from recent research efforts focused on developing both supervised and unsupervised machine learning algorithms to integrate single or multimodal patient-level omics data to robustly identify disease-relevant subpopulations. Upon reviewing the literature, we selected two classes of algorithms, similarity network fusion, SNF, and latent variable models, LVMs, for further investigation. Similarity network fusion, originally developed by the Goldenberg Lab, identifies subpopulations of patients by computing similarity between patients from either single omics, for example, transcriptomics, or also multi-omics. SNF includes an additional step that consolidates these omics-specific similarities into a few similarity via nonlinear kernel diffusion. On the other hand, latent variable models, such as independent component analysis, provide interpretable and lower dimensional representation of omics data, and thus enable the identification of disease mechanisms. Nevertheless, both algorithms lack the inherent ability to define distinct patient subpopulations, requiring additional downstream and model-appropriate clustering steps to identify patient subpopulations. This led us to the application of different combinations of feature transformations with clustering algorithms. For feature transformation, we tested no transformation at all, working directly on the pre-processed gene expression data, as well as latent variable models, including ECA, SNF, and also latent variable models like ECA prior to SNF. In terms of clustering, we tested community detection, spectral clustering, and we compared those against the k-means. We applied these multiple methods, mixing and matching omic feature transformation and patient stratification on multiple datasets. To ensure an fair and comprehensive testing, we selected both simulated and real-world patient-level transcriptomic datasets from multiple oncological indications. We assessed performances by quantifying the recovery of clinically relevant subpopulations that we defined as the agreement between inferred and oracle, ground truth patients and groups with the adjusted rate index as a metric. As oracle patient subpopulations, we use either built-in ground truth for simulated datasets or established molecular subtypes with clear, distinct phenotypes, prognosis, and clinical management for patient datasets. For example, a PAM50 subtyping for breast cancer. Moreover, we evaluated the stability of patient assignments to subpopulations across algorithms by quantifying the coefficient of variation of the adjusted run score across multiple bootstrap model runs where patients were randomized. While performance of dataset and context dependent, we identify some overall patterns, such as SNF and methods applied on LVM factors tend to perform better than methods applied directly on the data. Methods used to identify patients and groups impact the confidence of the performance in terms of stability. We need also to be mindful that performance are generally better in simulated datasets compared to real-world datasets. There are more details in the algorithms in the poster if you're interested, as well as an in-depth application in a disease context, where we found that the patients and groups track well with clinical endpoints and non-disease relevant mechanism signatures. This led us to a second use case, patient endotyping, where we are interested in identifying mechanism-specific subpopulation of patients by applying selected algorithms on features specifically associated with a pathway of interest, rather than the whole feature space. Again, the poster includes more details if you're interested, including an example workflow in breast cancer, as well as the application of a disease of interest, where we applied SNF followed by spectral clustering on selected latents, encoding a specific mechanism, inflammation in that case, in a TCGA cancer, and we could recover 2 patients' endotypes with the expected expression of macrophages regulation. Currently, we are working on tuning latent feature selection through several criteria, such as pathway enrichment and association with clinical phenotypes. And we are also constructing robust ensembles of features that are reproducible across different datasets. And this will provide the ability to further refine mechanism-specific patient subgroups and recover endotypes with greater clinical relevance for therapeutic intervention. Again, the detail of the implementation are found in the poster. Thank you for listening, and if you have any question, please come and talk to me when I'm at the poster or send us an email to get in touch. We are currently hiring in all our offices in London and New York in US. If you're interested in our mission, have a look at our jobs on our career page. Thank you.